Hi, hi everyone. I am Mark Shulowitz. I'm from GenScript, and it is my pleasure to moderate our next presentation, which is on a novel bacterial antiviral defense system. Our speaker for this presentation is Dr. Samuel Sternberg. Sam earned his BA in biochemistry from Columbia University and his PhD in chemistry from the University of California, Berkeley, where he worked under the mentorship of Nobel laureate Jennifer Doudna. After a brief postdoc and book writing stint, Sam joined Caribou Biosciences, which was a Bay Area startup at the time and still around, focusing on genome engineering, which was co-founded by Dr. Doudna. At Caribou Biosciences, Sam was a scientist and group leader of technology development. He then made the return to academia and also the return to Columbia University, where he joined the Department of Biochemistry and Molecular Biophysics, where he is currently a Sloan Research Fellow in Chemistry and a Pew Scholar in the Biomedical Sciences. Sam's doctoral and postdoctoral research focus on nucleic acid targeting by CRISPR-Cas and on the development of these systems for genome editing applications. His lab currently studies CRISPR-Cas biology with an eye towards applying their mechanistic knowledge for tool development. As you will hear in this presentation, his lab's work also extends beyond CRISPR-Cas9 to other interesting RNA-based bacterial defense systems. Sam is also a passionate advocate for science communication and outreach. He regularly presents to public audiences on the discovery of CRISPR-Cas immune systems and CRISPR-based gene editing, including a TED Talk and a book that he co-authored with Jennifer Doudna. Unfortunately, due to scheduling conflicts, Sam could not be here in person, but one of the members of his lab and a key contributor to the work that will be discussed, Stephen Tang, is with us to answer questions following the presentation. Stephen is an MD PhD student in Sam's lab. He has a BS in molecular biophysics and biochemistry from Yale University. His thesis work in the Sternberg lab focuses on reverse transcriptase enzymes from their biological role in antiviral defense pathways to their engineering for potential technological applications. I'm very excited about this presentation, and I think you will enjoy it very much. Bacterial antiviral defense systems have been a treasure trove for the discovery and development of gene editing tools, such as CRISPR-Cas. Most bacterial defense systems target foreign DNA for cleavage and destruction. Sam's presentation introduces a novel system based on reverse transcriptase that uses gene synthesis as an antiviral defense. Once again, if you have any questions during the presentation, please type them into the Q&A box. Following the presentation, we will address any questions. I now give you Dr. Sam Sternberg.
Hi, my name is Sam Sternberg, and it's my pleasure to present new unpublished work from my lab to you today. So by way of introduction, in the Sternberg lab, we're really motivated to understand how bacteria and other organisms defend themselves against viruses. We really started focusing on CRISPR-Cas systems. Over the years, that's got us interested in mobile genetic elements such as transposons. And then we've now been starting to study antiviral immune systems that have repurposed enzymes from transposons to deploy in the course of an antiviral response. And so broadly, one of the cross-cutting themes of our lab is RNA biology, as well as genes that have been repurposed over evolutionary timescales. And finally, how we can use some of the learnings from these biological areas to develop new genome engineering technologies. And so as a PhD student, I really entered this field with my fascination of CRISPR-Cas systems and the way that they use non-coding RNAs to defend many different bacteria against viral infections. You can see here in these captivating micrographs an E. coli bacterium on the left being bombarded with dozens of viral particles that will lyse the cell if the cell fails to defend against this infection. Over the years, what we've learned is that rather than this very simple view of the antiviral arsenal of bacteria shown in this schematic on the left, where about a decade ago, we just knew of about a half a dozen different families of immune systems. In fact, bacteria encode hundreds of immune system types, many of which have been discovered just in the last 5 or 6 years. Now, not all cells, of course, contain all of these, But if you look across all sequenced genomes, we can see a shocking diversity of immune system repertoires. This has revealed surprising new evolutionary links between both bacterial and eukaryotic antiviral immunity, showing how some of our own immune pathways in humans can be traced all the way back to bacteria. And finally, we, of course, see many examples of enzymes at this immune interface that can be deployed or repurposed for genome engineering technologies. And we just need to look at both CRISPR and restriction modification, the most common immune system in all bacteria, to see how revolutionary these DNA cutting enzymes have been for biotechnology. Now, on the right here, you're seeing a classification of immune system types by their abundance in bacteria. And what I want you to appreciate here is that the most common immune systems, the biggest blocks, are colored in green because they all fall into the category of immune systems that target and degrade viral DNA. Restriction enzymes and RM systems target and cleave viral DNA. And of course, CRISPR-Cas systems also use guide RNAs to target and degrade viral DNA during infection. The other observation I'd like you to make is that many of these systems are in gray because we actually have no idea how they work. They're unknown molecular mechanisms. And so the talk I'm going to share with you today is focused on one of these categories in gray called DRT for defense-associated reverse transcriptase. This was a family of systems that really captivated our attention a number of years ago. Now, just as a very brief refresher, reverse transcriptases, or RTs, are RNA-dependent DNA polymerases that use an RNA template to synthesize DNA, going in the opposite direction as the normal flow of genetic information in the cell. RTs are widely present across all three domains of life. So in retroviruses like HIV, the reverse transcriptase is essential to convert the RNA genome of the virus into DNA prior to integration into the host chromosome. Telomerase, our own mechanism of solving the DNA end replication problem, is a reverse transcriptase. And then most of our genome comprises retrotransposons, such as line 1 elements, that spread across our genome using reverse transcriptases. And then, of course, in the genome engineering space, one of the latest tools to add to the CRISPR toolkit, prime editing, relies on Cas9 fusions to reverse transcriptase. If you've ever used lentivirus to make DNA insertions, these, of course, also rely on those same retroviral RTs. And new genome engineering tools, such as work from Kathy Kahn's lab, use non-LTR retrotransposon-derived reverse transcriptases for gene insertion. And so because of this kind of confluence of interesting biological and biotechnological interest, we really wanted to set out to understand how these DRT immune systems function. And one of our driving curiosities was, as opposed to CRISPR and restriction modification that function by cleaving or degrading DNA, how would an immune system that synthesizes DNA be protecting the cell? This was really the driving question we wanted to answer. And moreover, if we look at the diversity of DRT immune systems, they fall into many different classes that differ in the specific genes in the operon, as you can see here in the exemplary operons for DRT families 1 through 9. And for all of these, the RNA substrate for the RT wasn't known. We certainly knew nothing about what cDNA the reverse transcriptases synthesize. And then finally, how would any of this provide immunity during viral infection? So these were really the questions that Stephen Tang, an extremely talented MD PhD student, set out to answer at the beginning of this project. How do these DRTs provide immunity from bacterial viruses? And you'll have a chance to ask questions directly to Stephen during the Q&A session after the talk. So at the beginning of this project, we focused on a family of systems called DRT2, using a system that had been first described in a publication from Feng Zhang's lab, derived from Klebsiella pneumoniae. This operon encodes one small gene, annotated in the figure, as well as an upstream non-coding RNA that have been detected in RNA sequencing data sets. Stephen encoded this reverse transcriptase on a plasmid that he then transplanted into E. coli and came up with a very unique approach to pull down flag tag reverse transcriptase and then isolate either the RNA or any DNA co-purifying with the reverse transcriptase, and then detect those ligands through high throughput DNA sequencing. Now, looking at the RNA component is a pretty standard workflow known as RIP-seq. But for the DNA workflow, Stephen came up with a new term, cDNA immunoprecipitation and sequencing, or cDIP-seq, to refer to this way of studying the reverse transcriptome of this enzyme. Here's some representative data where from the RIP-seq, you can see in the very top right of the graph that the most highly enriched RNA by far was in fact a non-coding RNA encoded directly upstream of the reverse transcriptase gene. But what was even more exciting was the cDIP-seq data, where now we saw this beautiful signature for cDNA deriving from that same exact non-coding RNA locus, proving that this reverse transcriptase is using that non-coding RNA in the cell to produce a complementary cDNA product. Now we can look at what these reads look like by mapping them to the reference locus. So here on the bottom, you can see the reverse transcriptase gene on the far right and a zoom-in view of the non-coding RNA. The RIP-seq data shows good coverage across the full length of the non-coding RNA, which we independently verified by 5' and 3' transcript start and end site mapping. When Stephen tested an inactive or mutant reverse transcriptase that I'll refer to throughout as YCAA, it's inactivated in the catalytic residues, we found that the RNA that the enzyme co-purified was exactly the same. And now when we look at the cDIP-seq signal, you can see a couple things. First of all, the reads here mapped to the reverse strand or the bottom strand, which is why they're plotted below the axis, and they corresponded to only a portion of the non-coding RNA, which is annotated at the bottom in pink. And now in the same control where Stephen substituted the inactive YCAA mutant, you can see that there's no presence of cDNA being made here, indicating that this product is indeed requiring an active RT enzyme. And for the rest of the talk, I'm just going to ask you to pay attention to the colors. Dark and light red are going to be the top and the bottom strand, because the strandedness is going to become quite important for the rest of the talk. Now, we looked more carefully at this non-coding RNA. So here you're looking at a predicted secondary structure, and colored in black and pink are the regions of the non-coding RNA that correspond to the scaffold structured elements at the 3' and 5' ends that we think are important for specific binding to the reverse transcriptase, and the template in pink, which is the sequence that is actually used for reverse transcription into DNA. And so we think the enzyme is being loaded in the middle of the molecule. It's reverse transcribing around this ring shown in pink to produce, as you can see on the right, a single-stranded cDNA product that corresponds to just a portion of the non-coding RNA. And support for the secondary structure came from covariance modeling that Dennis Zhang did to show that if we looked across about 300 homologous immune systems, they all shared a very conserved secondary structure. Now, next question we asked, does the cDNA product being made by this reverse transcriptase change during viral or phage infection? So here I'm showing now total DNA sequencing. We use a phage or a virus called T5. And so in the absence of T5, you see just a little bit of DNA from the bottom strand, but this is massively amplified in the presence of a phage infection. And intriguingly, what we found is that not only do we see cDNA corresponding to the bottom strand, but there was an equivalent increase in the same sequence on the top strand, suggesting that the DNA is becoming double-stranded during viral infection. Those data are quantified on the bottom. In the presence of the wild-type DRT2 plus T5 phage, you see a large increase in both bottom and top strand cDNA counts, and this is absent in the YCAA controls, which is an inactive reverse transcriptase. So from this, we could update our model to indicate that after the initial synthesis of the cDNA, the presence of a viral infection somehow triggers second-strand synthesis, yielding a double-stranded DNA molecule. Now, from some experiments that I'm skipping over, we had some puzzling results that suggested there was something more to the DNA products being made by this reverse transcriptase. And Stephen had a really important discovery by looking carefully at the raw sequencing reads in the cDIP-seq dataset. And in particular, what he found is that these reads were actually being clipped by the mapping algorithms because they had extensions both 5' and 3' of the annotated cDNA region that were not complementary to the reference genome he was mapping to. And so the algorithm just clipped these because they didn't match the reference, but this told us that the reverse transcriptase was making molecules that only matched the template across a portion of the cDNA. And in fact, these extended reads constituted the vast majority of the data set, indicating that this was the majority product. So Stephen next asked, what are these extensions that don't match the reference? And what he found is that they all come at the beginning of the same molecule. In other words, the reads that extend past the 3' end all match back to the 5' end. What this told us is that these enzymes are making concatemers of the same cDNA sequence stitched together head to tail. To further confirm this, Stephen changed his analysis, where now he analyzed the data looking specifically for bizarre reads that span the junction between two concatenated repeats. And he found that these concatenated repeat reads were in fact strongly amplified only with the wild type enzyme in the presence of viral infection, as you can see from the bar graph on the right. Now, these data came from short-read Illumina sequencing. We wanted to provide more evidence for this unusual reverse transcription behavior. And so we switched to a method that would both use long-read sequencing and obviate any dependence on PCR amplification during the library preparation. This work was led by George Lampe in the lab. He took phage-infected cells, extracted double-stranded DNA, and I used a library preparation workflow that, again, had no PCR steps, and then subjected these molecules to long-read sequencing by nanopore. And what he found quite remarkably is that these reads spanned from hundreds to many thousands of base pairs, and these thousand base pair long reads were the same exact 120 base pair repeat sequence stitched together over and over and over again, indicating something that was quite unprecedented for us, which is that this reverse transcriptase is cycling through the same exact template, making the same 120 base sequence over and over and over again. And so if we think about what this might look like if we could watch the enzyme in action, once it gets through one turn or one repeat length of the cDNA, somehow it jumps across to reinitiate synthesis on the beginning of the template, and then continues repeating this process over and over, yielding a mechanism that we call rolling circle reverse transcription. And we use this term because it's quite similar to a very widely studied phenomenon for plasmid and phage replication called rolling circle amplification. What's different is rolling circle amplification uses circular DNA templates. These reverse transcriptases are using non-circular RNA templates and instead exploit this really remarkable template jumping mechanism of the reverse transcriptase. So here's our updated model where the non-coding RNA is being used to produce not a short cDNA, but very long cDNAs that have the same sequence repeated over and over. During phage infection, this molecule is converted into double-stranded DNA. And then what's next? Why would a cell want to make concatenated double-stranded DNA upon viral infection? And how would this possibly lead to immunity? So the next breakthrough came from another really clever piece of science led by Stephen, where he found that this DNA was actually serving itself as a template for transcription into a concatenated RNA that had the same repetitive sequence as the DNA. And importantly, this concatenated RNA is the reverse complement to the non-coding RNA. And you'll notice that it's the bottom strand shown in pink, as opposed to the top strand non-coding RNA at the top, shown in dark red. And I won't go into the details, but we found promoter sequences at the junction between two repeats that explained why these molecules were being transcribed. Now, let me show you the data supporting this. In the top left of the slide, you can see RNA-seq data in the absence or presence of T5 phage. And only in the presence of phage did we see a huge increase in the amount of bottom strand RNA corresponding to the exact same region of cDNA. On the bottom left, Remonte used reverse transcriptase quantitative PCR to show that this RNA molecule was amplified 10,000-fold in the 10 to 20 or 40 minutes after viral infection. And then using northern blotting together with Luke Berkowitz, we found that these RNAs were extremely heterogeneous in size, spanning hundreds to many thousands of bases. And this size distribution was an excellent agreement with the large heterogeneous size distribution of the DNA molecules because of the rolling circle reverse transcription. So what would be the function or purpose for the cell to now make both the concatenated double-stranded DNA and then transcribe that back into RNA? So the answer to that question came from Stephen's third eureka moment on this project, where he was staring at this 120 base sequence for a long time and made the intriguing observation that one of the three reading frames lacked any stop codons. You can see here on the top 3 different potential reading frames. 2 and 3 have 1 or more stop codons. The first frame has none. And moreover, when he looked at what products would be made using this rolling circle reverse transcriptase, because each repeat length is exactly 120 base pairs, you would never introduce any in-frame stop codons across these long concatenated molecules. And so this suggested the possibilities that these might actually be functioning as open reading frames and be getting translated into protein. So this was a really provocative but kind of unexpected hypothesis. And so we really wanted to push ourselves hard to collect the necessary evidence to support it. I'm just going to give you a brief flavor, although more of the evidence is in the preprint. But Stephen used genetics approaches where he introduced silent, synonymous, non-synonymous, or missense mutations into the non-coding RNA that would be predicted to change the coding sequence of the resulting protein. And using phage infection assays shown on the top right, only the mutation to the stop codon on the bottom led to a loss of defense that resembled the same absence of defense as the empty vector control shown on the top. And he performed a number of other mutational perturbations, all of which supported this conclusion that this sequence is functioning as a gene that is translated into protein. So we could update our model that the end of this pathway is translation of the concatenated RNA to produce a protein. Now, because of this interesting phenomenon of generating concatenated DNAs that are functioning as genes, we refer to these as never-ending ORFs, or NEO, and the resulting protein as the NEO protein. We wanted more direct evidence, so we teamed up with Marko Jovanovic's lab to use mass spectrometry to unambiguously detect the NEO protein in phage-infected cells. You can see those data in the bar graph here. And we also showed that these were being made at a relatively high abundance. And so finally, why make NEO? What's the function or the antiviral point of producing this protein? I'm going to give you the punchline, which is that we had clues suggesting that NEO might drive growth arrest and that arresting the growth of cells would itself protect them from the viral infection. Some of the data to support that came from experiments performed by Valentin to separate all of the reverse transcriptase business from just neo, where we could express recombinant neo from a plasmid and then study the effect of neo on growth. And we tested different repeat lengths of that 120 base pair sequence, and we found that 1 repeat, no effect, 2 repeats, no effect. But anytime we tested 3 or more repeats, the wild type neo sequence had a strong arrest of growth, as you can see from the blue trace on the far right, but a scrambled control had no effect whatsoever. And we have additional data supporting this in the paper. So our model then for how this immune system works is that the reverse transcriptase is producing these very unusual rolling circle reverse transcription products. These are turned into double-stranded DNA only during a phage infection, and those DNAs get transcribed and translated to produce the neoprotein that arrests growth of the cell and thereby protects the cell and the broader population from the virus. Now, we have lots of questions that are still unanswered. First and foremost, how is neo doing this? We really want to understand the molecular mechanism of growth arrest induced by this protein. We'd like to understand why phage or viral infection catalyzes the production of the second strand of the DNA. And then going back to the diversity of these immune systems, just like CRISPR diversity, are these other flavors of DRTs using similar or different mechanisms for defense? And then I just want to end with some of the implications of our findings. So first of all, the neo gene escaped any detection by automated ORF algorithms because it's far too small to be detected, and it requires this unusual concatenation by the reverse transcriptase to attain its final form. And so this has challenged the way that we actually look for genes. Secondly, the neo genes are not encoded on large chromosomes, plasmids, genomes. They are free-floating molecules that are only produced transiently, challenging the conventional notion of how genes get encoded, stored, and inherited. Third, this really changes the way that you can stitch together genic information rather than always being in a linear, one-dimensional form. This gene is produced by unusual topologies of sequences concatenated head to tail. Fourth, unlike the typical flow of genetic information from DNA to RNA to protein, DRT2 requires multiple conversion steps between DNA and RNA, back to DNA, and then to RNA before the protein can get made. This work really highlights the versatility of RNA that can be both a functional molecule and a carrier of genetic information. And then finally, what we're really excited about is to think about creative ways to exploit these unusual reverse transcriptase activities for new kinds of genome engineering tools. And so with that, we're really hunting for other kinds of cryptic genes that might be hidden in the genome that have escaped detection thus far. And I'll end by just highlighting again the preprint that Stephen led. And again, please ask your great questions on this project to him during the Q&A. And then a big thanks to the entire Sternberg lab, as well as our sponsors and all of you for your attention. Thank you very much.
Great. Well, thank you so much for your patience in that.
Sure.
I always like to tell people getting connected here is half the battle, so we did good.
So I don't know how to get over this because I hear the other.
Well, thank you, Sam, for an interesting and really exciting presentation. That was fantastic. So we will now open the floor for questions. So if you have any questions, please enter them in the Q&A box. We've got some questions already. And just as a reminder, Stephen Tang, the student from Sam's lab, will be here to answer your questions. So if you have any technical questions, you can also message us through the chat box or the help desk. So let me start in on the questions. So hi, Stephen.
Hello.
So the first question that we have is, what do we know about the length of the neoproteins? And do we know anything about the mechanism of translation termination for a stop codon-less gene?
Yeah, it's a good question. So we don't know a lot about the length. We can only infer what the length is based on the northern blot that Sam showed in the presentation, showing the neo-mRNA being highly heterogeneous and in some cases very long. Again, the process of making the neo gene is likely governed by mostly stochastic fall off of the reverse transcriptase from the template. And so what we're thinking is that the reverse transcriptase will make the cDNA up to however many repeats until it falls off. And then that will serve as the template for transcription, which will then serve as the template for translation. And the resulting protein product then could be any number of repeats long. We show that you only need to be 3 repeats long in order to have this growth arrest phenotype, but it could be as many as 40 repeats long, which is what is suggested by the nanopore sequencing that Sam showed. So that's what we think in terms of its length, just that it's probably a range of lengths, at least 3, so that it can have the growth arrest phenotype. And then in terms of the translation of this stop codon-less gene, yeah, that's actually another really interesting aspect of this, because in bacteria, there are mechanisms, quality control mechanisms that basically get rid of any mRNAs that don't have stop codons. And we found in some mass spec experiments that are in our preprint but not shown in this presentation that actually one of the most upregulated pathways in cells infected by phage that express DRT2 is this alternative ribosome rescue pathway that would allow these stop codon-less mRNAs to actually produce functional proteins rather than being tossed out.
Okay, thank you. And that's a reminder for our audience that there is a preprint available, so definitely check it out. Go through some more of these questions. So do you know how many potential neo sequences or neo proteins may exist?
So we did take one sort of broad bioinformatic approach in our pre-printed work to get at this question. Of course, we were excited to see that it exists for one system, but when you have such an unusual result, it's even more exciting if it's something that exists broadly. And so we took a few hundred homologs of DRT2 and bioinformatically predicted whether we could find the sequences associated with those DRT2 homologs that would enable the production of a neogene based on what we know from the KPN homolog that we experimentally studied. And from that analysis, we have on the order of 200 predicted neo sequences. Out of those 200, we went and actually experimentally confirmed that 5 of them have the same function in E. coli that the KPN homolog does.
Okay, cool, thanks. And we have a question on the secondary structure of the non-coding RNA. So is anything known, is the secondary structure important for that non-coding RNA? And if it is, would it be possible to optimize the secondary structure to improve reverse transcription?
Yeah, so this also wasn't in the talk, but it's in our preprint. We made a number of different perturbations to the non-coding RNA, which had a range of effects. So the smallest perturbations involved changes of single base substitutions. Those, for the most part, seemed pretty well tolerated. But when we made larger perturbations by, for example, mutating entire stem loops so that we were abolishing base pairing of those stem loops, you could see a complete loss of rolling circle reverse transcription activity in some cases, but then in other cases, a complete retention of it. So it basically seems like there are some stem loops which are critical for reverse transcription and others which are dispensable, which adds to our idea that you might be able to rationally engineer the non-coding RNA to produce a custom cDNA sequence based on knowledge around which stem loops are actually critical.
Okay, great, thanks. I have kind of a question about if there's any potential off-target effects from these reverse transcriptase. So how do DRT2 systems differentiate between the host non-coding RNA and foreign nucleic acids to initiate rolling circle reverse transcription? And what implications might this have for the specificity and potential off-target effects of these systems in bacterial antiviral defenses?
Yeah, so again, going back to this ties in well nicely to the secondary structure question, which is that there appears to be a pretty strong dependence on the non-coding RNA structure for reverse transcription. And if you remember back to the part of the talk where Sam was distinguishing the template region and the scaffold region of the non-coding RNA, the perturbations that we tested that involved mutating the scaffold region largely abolished any reverse transcription activity, whereas the mutations to the template region were better tolerated. So we think likely what's happening is there's a scaffold where the structure is really important, and that's likely how the RT even finds the cDNA synthesis start site to begin with, is by binding to this structured non-coding RNA, and that's likely how it distinguishes its cognate template non-coding RNA from just other RNAs floating around in the cell. That there's this really important binding interaction between the RT and the non-coding RNA scaffold that allows it to actually then begin reverse transcription.
Okay, thank you. I think we have time for one more question. So the last question that we have time for. So I think Sam might have touched on this at the end of his talk, but are you exploring the potential molecular trigger for this mechanism? So in other words, how are phage sensed? And sort of speculative, but do you think this is universal for all phage, or different phage might have different triggers?
Yeah, it's a good question. So the interesting thing about phage bacteria interactions is that they tend to be highly specific, where there tends to be a limited specificity of, or I guess a narrow specificity of phages for a given E. coli host. So for the most part, we wouldn't expect the system to have extremely broad activity against a bunch of different phages, just because the situation that it evolved with was a rather specific host phage specificity. But what I can say is that we do have some pet hypotheses around what might be triggering the mechanism, which are that given that the second-strand synthesis step, where you convert the single-stranded cDNA to a double-stranded cDNA, seems to be really critical for initiating the cascade of events that lead to neo production, we think that the question of what polymerase is actually performing second-strand synthesis could be really, really critical. And to that, we don't have a concrete answer. But if it's the RT itself that is performing the second-strand synthesis, then there could be some interaction with, for example, a protein encoded by the phage that changes nucleotide levels in the cell that will govern, that will change the activity of the RT. Or pet hypothesis that we're really intrigued by is the idea that it's actually a phage polymerase, which is generating the second strand, in which case that would really nicely explain how there's this controlled production of just a single strand in the absence of phage, but then it gets completely turned on in the presence of phage.
Okay, wow. Well, thank you so much, Stephen, for answering these questions. I think, any great discovery, this opens up a whole bunch of questions, and there certainly are a lot opened up here, and I'm looking forward to seeing where this research takes you. I'd like to thank all the participants for joining us today, and don't forget to visit our booths in the exhibit hall, and stick around for our next session. This is only the second one today, and we have a whole bunch of exciting sessions. So thank you, everyone.
Thank you.